Good day, everyone. Thank you for joining us for our live webinar, which will be presented today by our guest speaker, James Olzmann, entitled The Cellular Lipid Homeostasis from Lipid Droplets to Ferroptosis. My name is Maria Fedorova, and I'm based at the Technical University of Dresden, and I will be your host for today's webinar. This webinar is kindly sponsored by Cayman Chemical. The webinar will be around 45 minutes, allowing for 10-15 minutes for the questions and discussion at the end. And now I'm very happy to introduce our speaker for today. So Dr. James Olzmann is a Doris Calloway Chair and Professor of Molecular Therapeutic at the University of California, Berkeley, with joint appointments in the Departments of Molecular and Cell Biology and Nutritional Science and Toxicology. James received his bachelor degree from the University of Michigan and PhD from Emory University. He conducted his postdoctoral research with Dr. Ron Capito at the Stanford University before joining Berkeley in 2013. James' lab is investigating the mechanisms that regulate cellular lipid homeostasis, focusing on lipid droplets and ferroptosis. His group seeks to uncover how cells maintain lipid balance and to identify new therapeutic strategy targeting lipid metabolism and oxidative stress and cell death. His contributions have been recognized with honors including the Presidential Early Career Award for Scientists and Engineers, the ACCB Günther Blober Early Career Award, the ASBMB Avanti Lipid Award, and appointments as Chan Zuckerberg BioHub investigator and Miller Institute professor. So we are all very excited, James, for your presentation. So please share your screen and the stage is yours.
Okay, so first off, I just want to say thank you to Lipid Maps for organizing this terrific webinar series and to Maria for the invitation and kind introduction. It's a real pleasure to join you today to tell you a little bit about some of our ideas and recent findings related to the cell biology of lipid homeostasis. So my lab is interested in cellular lipid metabolism. We're interested in very basic fundamental questions about the cell biology of lipid metabolism, as well as how this goes awry in different diseases and how we might begin to develop therapeutics to target these processes. We're interested in ER-derived neutral-lipid storage organelles called lipid droplets, how these organelles form, how proteins target these organelles, and how proteins are degraded from these organelles to remodel the proteome under different conditions, for example, by the ubiquitin proteasome system. We're also interested in how lipid droplets make contact with other organelles and the composition and functions of these membrane contact sites, for example, to regulate signaling or the transmission of lipids to organelles within the cell. In addition, we're interested in very broadly in the mechanisms of lipotoxicity and what we like to refer to as lipid quality control, or the processes within the cell that regulate lipid damage and the subsequent toxic effects of lipid damage, for example, cell death, known as ferroptosis. So today I've separated this webinar into two sections based on two overarching questions that we're interested in the lab. The first is, how is lipid quality maintained? And so here I'll focus on some of our recent findings related to the damage and oxidative damage to stored neutral lipids within lipid droplets, as well as our development of small molecules to target these processes. And in the second portion of the talk, I'll switch gears to focus on the formation of lipid droplets. So how are lipids stored in lipid droplets? And so I'll focus on some of our recent genetic screens, looking at ER neutral lipid flux and the regulation of lipid droplets, as well as some surprises that have come out of these genetic screens that were not anticipated. So our cells have evolved highly sophisticated quality control systems to combat the accumulation of damaged biomolecules. And this is depicted here in this conceptual schematic. You can imagine very simply that you could have some sort of damage-sensitive macromolecule, and that this damaged product could be toxic. It could result in impairments in cellular fitness or the initiation of cell death pathways. Fortunately, we have quality control systems that suppress the accumulation of these damaged products, and these fall into a few different classes. We have preemptive quality control systems that suppress damage. We have repair quality control systems that repair this damage. And in some cases, we completely degrade that damaged product, or we convert it into a benign product. I think this is very well exemplified from decades of research in the protein quality control field. For example, you can have misfolding of proteins due to mutation or damage, like oxidation, but we have chaperones that can suppress this misfolding or refold these proteins under some conditions. We have systems like the ubiquitin proteasome system or lysosome autophagy system that degrade these proteins. And we also have homeostatic stress responses that sense this damage and initiate homeostatic responses to hopefully achieve balance within the cell. Now, our lab is very interested in lipid quality control, and I think we can think of it in similar concepts, that lipids also can be damaged. For example, we know that polyunsaturated fatty acids are prone to oxidation, and we're interested in the quality control processes for these lipids. How could this be suppressed? How could this damage be repaired? And in some cases, how could this damage perhaps be degraded or converted into benign products? And this interest in lipid damage and lipid quality control led us to become interested in a form of cell death that many of you are probably familiar with, that is known as ferroptosis. And this name was first coined by Scott Dixon and Brent Stockwell back in 2012, and we've seen really a lot of growth in the field in the last decade. Now, this form of cell death attracted us because for us, this is really a regulated form of cell death that's due to the failure in lipid quality control. So in this form of cell death, we have the accumulation of oxidatively damaged membrane lipids, and this can eventually lead to membrane rupture in cell death. So we have the incorporation of oxidative sensitive polyunsaturated fatty acids, or PUFAs, into these phospholipids. And we have the production of reactive oxygen species that eventually lead to the generation of these membrane lipid peroxides. Now, there are well-known quality control systems, for example, mediated by the glutathione-dependent peroxidase GPX4, which utilizes glutathione to convert these lipid peroxides into non-toxic lipid alcohols. So this would fall into the class of degradation or conversion of that toxic lipid into a more benign species. There are a variety of different small molecules that can be used to trigger and study ferroptosis, developed by many groups. These are some of the first developed by Brent Stockwell, RSL3, which is an inhibitor of GPX4, and Erastin, an inhibitor of system Xe- that blocks cysteine uptake and the synthesis of glutathione. So we've really seen a huge growth in this field in the last 5 to 10 years due to the potential therapeutic importance of ferroptosis, with the idea that you could potentially trigger ferroptosis as a way to kill drug-resistant cancer cells, or that you could inhibit ferroptosis as a way to ameliorate some of these diseases associated with cellular degeneration, like ischemic tissue damage, neurodegeneration, and others. Now, when we first became interested in ferroptosis, we noted that certainly in many cancer cells, you can inhibit GPX4, and these cells would die through ferroptosis. But we also noted that many cells were resistant to inhibition of GPX4. In some cells, we could knock out GPX4, and they would continue to grow and divide, at least in cell culture. And this suggested that there must be additional protective mechanisms that prevent ferroptosis. And so this gave rise to this question that was tackled by a former postdoc in the lab, Carol Berzuker, who's now a principal scientist at Calico. And he was interested in asking, what are these additional protective mechanisms that can promote lipid quality control and compensate for the loss of GPX4? So we did a synthetic lethal CRISPR screen. This was a number of years ago now, in a ferroptosis-resistant cancer cell line, U2OS. This is an osteosarcoma cell line. We introduced a CRISPR library and then simply treated with the GPX4 inhibitor RSL3. And you can imagine under these conditions, cells that are sensitized by loss of an additional protective factor would die, and they'd be disenriched in the final population, and we could identify these by deep sequencing. And And I remember when we first did this screen, I imagined that we were going to get out many hits. We had ways to deconvolve this and ways to put these into pathways and prioritize. When we did this screen, it was very clear what gene we should focus on. It had a high confidence score and a sensitizing effect, indicating that loss of this gene was sensitizing to ferroptosis. This was a gene, AFM2, which we renamed ferroptosis suppressor protein 1, or FSP1. And I just want to point out that this was co-discovered by Marcus Conrad and Pedro Angeli in a co-published companion paper at the same time. And our findings are in very strong agreement regarding the cellular roles and mechanism with Marcus and Pedro. And so just a little bit of data from this paper. We knocked out FSP1 in these cells. These cells will continue to grow in cell culture, but they're highly sensitized to the induction of ferroptosis with the GPX4 inhibitor RSL3 or with other ferroptosis inducers. So we see a classic morphology in which these cells have rounded up, their plasma membrane is ruptured and their cytoplasm is leaked out, and we're marking them with a cell death dye Cytox in this case. We can also do this in dose response, so in high amounts of RSL3, these cells are quite resistant, so very little cell death, but these are highly sensitized in two different independent knockouts of FSP1 with an EC50 here of around 150 to 100 nanomolar. So this paper was published now 5 or 6 years ago, so I'm just gonna summarize the mechanism that we discovered here so I can share some of our more recent discoveries and findings. So I had mentioned that we have this quality control system mediated by GPX4. Using this genetic screen, we identified FSP1 as a parallel quality control system, and it operates through a distinct mechanism. So while GPX4 is an enzyme that converts these lipid peroxides into lipid alcohols, FSP1 in an NADPH-dependent manner, it's an oxidoreductase, converts oxidized CoQ or ubiquinone to its reduced form, ubiquinol. And this functions as a very potent radical-trapping antioxidant that can suppress the propagation of these lipid peroxy radicals and lipid peroxides in the membrane. And so this was interesting both from the ferroptosis perspective, also from the mechanistic perspective, and it solved a long-standing question as to what CoQ is doing in these non-mitochondrial membranes, and we think it's there as a radical-trapping antioxidant to protect our membranes from damage. So we're cycling this pool of CoQ and protecting membranes from oxidative damage. So this raised several questions that I'd like to touch on today, one being this concept of subcellular lipid quality control. And I think this can be seen similar to protein quality control, that there are different mechanisms to degrade damaged proteins in different compartments in the cell, and this can have very different phenotypic consequences if you accumulate damaged proteins in these different organelles. So we imagined very similarly that we could conceptualize that damaged lipids could accumulate in different organelles, for example, lysosomes, peroxisomes, mitochondria, and that this could have very different mechanisms of regulation and very different phenotypic consequences. So I'll touch on this idea. And then we are also very interested in the therapeutic potential of targeting FSP1 as a therapeutic for cancer. So this idea of subcellular lipid quality control initially led us to lipid droplets, where we've focused on because of our interest in stored neutrolipids and their importance for many diseases, as well as our finding that FSP1 localizes very strongly to these organelles. And so we had seen this in our initial publication. This was what I'm showing you here in the images are a genetically modified cell line in which we knocked in a halo tag into the endogenous locus of FSP1. And we can see that this endogenously tagged FSP1 very nicely localizes to lipid droplets, marked here with BODIPI 493503, as well as to the plasma membrane, here marked with cell mask. So it has multiple localizations, and it's probably potentially regulating lipid damage at different locations in the cell. We also biochemically fractionated cells. We could float lipid droplets on sucrose gradients, and you can see very nicely that FSP1, a portion of FSP1, very nicely localizes to this lipid droplet enriched buoyant fraction. So this raised a question that's been addressed by a really talented current postdoc in the lab, Mike Longa, who's currently on the job market. And he's been looking at this process and asking this question regarding the stored neutrolipids. So are lipids stored in lipid droplets protected from oxidative lipid damage? And the prevailing hypothesis here being that FSP1 could potentially be localizing to these organelles to prevent the oxidation to these stored lipids. So a little bit of introduction into lipid droplets. These are unique organelles. They emerge from the outer leaflet of the ER into the cytoplasm, and they have a unique ultrastructure. So they have a single phospholipid monolayer that surrounds a core of neutrolipids. This is mostly triacylglycerol or triglycerides and fat, and cholesterol esters, though they can also sequester other types of hydrophobic molecules. They have proteins that decorate the surface that regulate their functions, and really these organelles function as cellular hubs of lipid metabolism. So they're really dynamic storage depots of lipids, and they can rapidly mobilize these stored lipids for energy, for example, breakdown of fatty acids by beta oxidation, for the synthesis of membranes or lipids, signaling molecules, and by sequestering these lipids, they play important roles in lipotoxicity. And not surprisingly, they're connected with a wide variety of human disease pathologies, some of which we're studying in the lab. And this idea of lipotoxicity also has been connected with ferroptosis as well. And so there's this emerging data in the field, which I think is quite strong, and the idea that polyunsaturated fatty acids, which I had mentioned are sensitive to oxidative damage, can be sequestered within these neutrolipids, triacylglycerol, and this can be protective by preventing the flux of those polyunsaturated fatty acids into membranes, like the plasma membrane. So there are many questions that remain here, how these are sequestered, whether oxidized PUFAs are sequestered within the droplets, where these are coming from, for example, phospholipase-released PUFAs or dietary PUFAs. But the prevailing idea is that these lipid droplets are protective. But I think there's a remaining question as to what happens to these neutrolipids once they're stored in the droplet. Are they prone to oxidation? What would the consequences of oxidation of stored neutrolipids be? Or are they protected, and how? And so these were some of the questions we wanted to address. And the approach we took was to initially look by using lipidomics strategies, and Mike performed a series of lipidomics experiments together with Maria Fedorova in Dresden. And I'm just going to present one of these here to you in this slide, which I really think exemplifies some of the key findings from this publication. So what Mike did is he loaded UTOS cells with fatty acids. These have relatively low lipid droplets under basal conditions. So we stimulated the formation of lipid droplets so we could look at the consequences for neutrolipids. And then he performed both untargeted lipidomics and semi-targeted oxidized lipid analyses. And what we were really struck by was by the oxidized lipids that accumulated in our FSP1 knockouts. So shown in kind of the yellow-orange circles here are oxidized triacylglycerol molecules that are accumulating in the FSP1 knockout. We're really excited by this finding because these are in FSP1 knockouts grown under basal conditions. So we're not treating them with any inducers of oxidative stress or with inhibitors of GPX4. And so that this really suggests that FSP1 is playing an important role in regulating and protecting stored neutrolipids from damage that's not compensated, or at least not completely compensated, by any other enzymes or proteins in the cell. We could also confirm that these oxidized lipids are accumulating in lipid droplets. So we did these imaging studies using Bodipi C11, which under oxidized conditions fluoresces in a unique wavelength so we can detect these oxidized lipids. So this is an indirect reporter of lipid oxidation. And what we saw is when we treated with an FSP1 inhibitor called FSN1, and I'll come back to this molecule in a few slides, is that we started to accumulate increased signal within these cells, and this signal was shown to be punctate and overlap with our lipid droplet marker, LipiBlue. And so we accumulate this oxidized lipid signal, and this could be blocked by known inhibitors of ferroptosis, radical trapping antioxidants like FER1. So we could detect the accumulation of oxidized triacylglycerol in these cells, as well as their accumulation within lipid droplets using this indirect reporter. Mike then went on to perform biochemically reconstituted assays. These are using artificial lipid droplets that are generated in vitro, similar to liposomes, but in this case we have stored neutral lipid, trioline, surrounded by a phospholipid monolayer. We can then use this Phoenix assay developed by Derek Pratt's group, in which we add a radical initiator, D10, to trigger lipid peroxidation, and then we can study lipid peroxidation using that fluorescence reporter, BODIPC11. This is a useful reconstituted system because then we can add purified FSP1 and different substrates to look at their functions. So what I'm showing you here is the accumulation of lipid peroxides over time using BODIPC11 as an indirect readout. We see that with no protein, we accumulate lipid peroxides here, shown in black. If we add in wild-type FSP1, together with its substrates NADH and CoQ10 or ubiquinone, we suppress the accumulation of these lipid peroxides. Eventually, we start to accumulate lipid peroxides again. We have this inflection point. And what this is we've consumed all of the NADH in the reaction, so we can no longer recycle CoQ. If we simply added more NADH, we extend this lag phase even longer. If we add an inactive version of FSP1, E156A mutant, this has no effect. So this was really good confirmation that in this reconstituted system, FSP1 together with CoQ could directly suppress lipid peroxidation. But because we were using an indirect fluorescence readout, it was unclear whether the neutral lipid, triacylglycerol, was being oxidized or the phospholipids in this system. So Mike performed in some additional lipidomics analyses of these experiments. I'm just showing you here the triacylglycerol. And what he shows is here that we can detect triacylglycerol peroxide, so it's being oxidatively damaged, and FSP1 together with CoQ and NADH can suppress this. Very similar to that lag phase we were seeing with BODIPC11. We could also detect oxidized phospholipids, so it seems like in this reconstituted system, FSP1 is able to suppress the damage to both stored neutral lipids as well as the bounding phospholipid monolayer. So these, together with the lipidomics and imaging experiments, strongly implicate FSP1 as a defense factor to protect stored neutral lipids in lipid droplets. We wondered about the phenotypic consequences of this, and I'm going to show you some cellular experiments that we've performed, but we're interested in now moving to physiological conditions where you might imagine this might be important. So you could certainly imagine this could be important in cancer cells with high amounts of lipid droplets, in fatty liver where they begin to accumulate neutral lipids and lipid droplets, or other conditions like glial lipid droplets and neurodegeneration. So in these conditions, we're using cancer cells again. We load with a polyunsaturated fatty acid to stimulate oxidation-prone lipid droplets. So this arachidonate's incorporated into arachidonate-rich triacylglycerol, and then we can look at the consequences of FSP1 inhibition. So in this system, we again perform lipidomics, confirming that FSP1 inhibition or FSP1 knockout leads to the accumulation of oxidatively modified triacylglycerol and neutral lipids, and that this can be blocked by radical trapping antioxidants like FER1 as expected. Now, if we added a MUFA, a monounsaturated fatty acid, which is less prone to oxidation, and looked at cell death, there was very little effect. This is a very good stimulator of triacylglycerol and lipid droplets, but we don't see any cell death occurring, maybe a little bit at 400 micromolar, these high concentrations, but no real difference of our FSP1 knockout. But we saw something quite different when we used our polyunsaturated fatty acid, our PUFA, arachidonate. As we get to higher conditions, this is well tolerated under wild-type cells up to about 200 micromolar, but we see something very different with our FSP1 knockouts. Here, as early as maybe 50 micromolar, we start to see the induction of cell death, and these cells are nearly completely dead by about 200 micromolar in the cell line. And this indicates that loss of FSP1 is protecting from PUFA-induced cell death, right? And so we wanted to understand whether this was connected with lipid droplets and the phenotypes we're seeing with oxidation to those stored neutral lipids. And to do this, we use DGAT inhibitors to block the formation of triacylglycerol and lipid droplets under these PUFA-induced conditions. And so you can see here, this is quantifying from imaging studies that we together when we add inhibitors of DGAT1 and 2, which inhibit the conversion of diacylglycerol to triacylglycerol, which is then stored in the lipid droplets. We strongly inhibit lipid droplet biogenesis, and we strongly inhibit triacylglycerol formation. This quantified from thin layer chromatography experiments. We then wanted to look at the effects on ferroptosis and the induction of cell death in our knockouts to see if those droplets were really the culprit here. So what we're seeing here is in our knockouts in the black bar is under treatment with those PUFAs, we see induction of cell death. This is what I also showed you on the preceding slide. If we treat with an inhibitor of ATGL, this is an enzyme involved in lipolysis and breakdown, we actually worsen cell death. And if we treat with our DGAT inhibitors, which block the biogenesis of lipid droplets, we completely suppress the cell death. So this really demonstrates that the formation of these PUFA-rich lipid droplets are required for this condition to trigger ferroptosis with the loss of FSP1. So let's put this together into a model to tell you about what we've discovered here. So what I told you in the first portion is that we found FSP1, and particularly an important role for FSP1 in suppressing ferroptosis at the plasma membrane through the generation of reduced CoQ, this radical trapping antioxidant. In this new study, we demonstrated that FSP1 at lipid droplets is important for protecting stored neutral lipids from damage. So not just phospholipids, but also stored neutral lipids like triacylglycerol. And this is operating through a similar mechanism. We've shown this both in cells and in reconstituted assays that FSP1 is recycling CoQ to generate the radical trapping antioxidant form to suppress the propagation of these damaged lipids. And eventually this propagates to cause ferroptosis. So I think this is really interesting for a few reasons. I think the conceptual take-home points here are that this indicates is a really good example of a compartment-specific lipid quality control mechanism in which FSP1 localized to lipid droplets is protecting this local environment of stored neutral lipids. And this also gets to this other concept that's emerged in the field, that ferroptosis can initiate at different intracellular membranes. So there are stories from different groups, like Brent Stockwell has shown that ferroptosis may initiate from the ER under some conditions, like induction with Avastin. Rafael Rodriguez and others have shown that ferroptosis may initiate at lysosomes under certain conditions. There are other examples from the mitochondria, and what I'm showing you here is from the lipid droplet. And I think these aren't mutually exclusive models, but really demonstrate the context-specific importance of the composition of the lipids as well as how we're inducing ferroptosis, in this case with FSP1 inhibitors. And I think a big question remains in the field of how the damage from these intracellular organelles eventually propagates to the plasma membrane to trigger ferroptosis. So this led us to become very interested in this pathway, as well as how we could maybe target this pathway as a way to kill cancer cells as a therapeutic. And this was work from a previous postdoc, Joe Hendricks, who's now a scientist at Vulcan Therapeutics, and Amalia Magriotti, who's a current postdoc. Can we target FSP1 with small molecule inhibitors? And we were motivated by studies in our lab, as well as emerging studies from many in the field. For example, just 2 months ago, there were these two studies, one from Thales Papagiannakopoulos and one from Jesselyn Ubelacker, in vivo, demonstrating that genetic deletion of FSP1 or small molecule inhibition of FSP1 could trigger ferroptosis and suppress tumor growth in these in vivo models, really motivating us to follow up on this pathway as a potential therapeutic mechanism. And I think what was really interesting about their two studies is that in vitro, we know that loss of FSP1 is relatively well tolerated. But what their studies show is that in vivo, the environmental context and tumor microenvironment sensitizes these cells, and loss of FSP1 results in a very nice tumor suppression. So certainly the in vivo context and tumor microenvironment are emerging as important regulators of ferroptosis sensitivity. So we also know from a collaboration with Michael Dupage and his graduate student at the time, Jesse Garcia Castillo, that targeting of FSP1 in non-tumor cells may also have therapeutic benefit. So what Jesse did in this experiment was he knocked out the gene encoding FSP1, AFM2, in T cells or in Tregs. And very strikingly, he saw that knockout of FSP1 in Tregs, the gene being AFM2, resulted in strong tumor restriction. And this was because this led to increased conventional T cells, which CD8 positive, which were then suppressing the tumor growth. And these together suggest that targeting FSP1, both in the tumor for triggering of ferroptosis, as well as for the regulation of the immune system, may both have therapeutic benefits. So these motivate us to look into the development of small molecule inhibitors. We did this together with Julia Shaletsky and Eddie Weary at the UC Berkeley Drug Discovery Center, performing a small molecule screen of 115,000 small molecules in an in vitro activity assay using purified FSP1 protein. And then we took hits from the screen and rescreened these in an orthogonal cell-based assay of ferroptosis. This led to the identification of multiple classes of FSP1 inhibitors. Some of these are shown here, and you can see the different chemical scaffolds, giving us quite a bit of chemistry to work with. We primarily focused on this first class of FSP1 inhibitor because these were the most potent straight out of the screening deck. With this molecule, FSEN1, or ferroptosis sensitizer 1, being the most potent and the molecule that we've characterized in the most detail. And among these initial classes of FSP1 inhibitors, this class, this core pharmacophore was conserved among those molecules, suggesting that this structure here is particularly important for its mechanism. We could inhibit FSP1 in vitro, showing that this is a direct inhibitor with an IC50 of around 300 nanomolar. And we could also inhibit FSP1 in cells, and I'll just show you one of these experiments here using KEAP1 mutant lung cancer cells, which are very resistant to the induction of ferroptosis. You can see that these cells continue to grow and divide with inhibition of GPX4 with RSL3, but if we co-treat these cells together with FSEN1, our ferroptosis inhibitor, we get very rapid and complete induction of ferroptosis, consistent with these having two parallel pathways that can compensate for each other. Now, at the end of this study, we had some mysteries remaining. One was the mechanism of how FSP1 was inhibiting, how FSEN1 was inhibiting FSP1. And we also noted that FSEN1 was specific to human FSP1, and it does not inhibit well mouse FSP1. And it was unclear on why this was the case. Recently, we've been able to answer some of these questions through a really nice collaboration with Da Jia at Sichuan University in China, where we solved the co-crystal structure of FSP1 bound to our inhibitor FSEN1. So here we see the oxidoreductase structure. This had been solved by other groups, and we see a similar structure here in our structure, coordinating FAD, 6-hydroxy-FAD, and NAD plus in this structure. And then we see our inhibitor here bound to FSP1. And importantly, this showed some key contacts. For example, it makes this pi-pi stacking interaction with phenylalanine 360. This also solved the mechanism, and that we also know that this is the substrate binding pocket for CoQ. So it seems that FSEN1 is a competitive inhibitor of FSP1, and likely this interaction with this pi-pi stacking interaction is very important. And indeed, in experiments I won't go into, we can mutate this phenylalanine to a leucine or an alanine, and we lose the ability to inhibit FSP1 with FSEN1. Importantly, also, this structure solved the question of why this is specific to human FSP1. Human and mouse FSP1 are highly similar, so I was initially surprised that this inhibitor did not inhibit mouse FSP1. But when we go back to the sequence of FSP1, we see that this phenylalanine 360 in human is actually a leucine in mouse FSP1. It's also a leucine in Drosophila FSP1. If we make a phenylalanine to leucine mutation, this blocks the ability of FSN1 to inhibit human FSP1. We can also do the corresponding mutation. We can take mouse FSP1, mutate that leucine to a phenylalanine, and we're able to gain inhibition. So humanize the mouse to FSN1. So this structure was very useful for many things. This was the first structure of FSP1 together with an inhibitor. It solved these questions around the mechanism of FSN1 inhibition of FSP1 and solved this question around the species selectivity. And it also provides us with a basis for rational medicinal chemistry and how we might start to improve this molecule to increase its potency as we move from in vitro to in vivo preclinical models. Okay, so in this first portion, I told you about our discovery and some of the progress towards therapeutic targeting of FSP1 and lipid quality control. Specifically, I told you how we use genetic screens leading to the identification of FSP1. We've identified a mechanism in which FSP1 is able to recycle radical trapping lipophilic antioxidants like CoQ. Recently, we've discovered a role for FSP1 on lipid droplets where it's protecting stored neutrolipids from damage, and eventually these can propagate and lead to ferroptosis. And we've also developed small molecule inhibitors against FSP1 and recently solved the co-crystal structure, enabling rational medicinal chemistry of these molecules, which I think will be beneficial as we start to move to more therapeutic models. Okay, so this is really focused on lipid quality control and lipid damage and how we protect stored neutrolipids from damage. I'd like to now shift gears to some of our genetic screens and understanding of how neutrolipids are stored in lipid droplets and how these organelles form in cells, and some surprises that have come out of these studies. So we've already talked about lipid droplets a little bit, but I'd like to give a little bit of a more depth into our understanding of the formation. So neutrolipids like triacylglycerol are synthesized at the ER. These neutrolipids, triacylglycerol and cholesterol esters, are deposited between the leaflets of the bilayer within the hydrophobic phase of this membrane. And then at a sapient containing lipid droplet assembly complex, this lowers the energetic barrier for phase separation of these neutrolipids, and these lipids form a lens structure, which eventually emerges from the outer phospholipid leaflet of the ER into the cytoplasm. There are proteins that can directly insert into the lipid droplet, and proteins that first insert into the ER and then traffic to lipid droplets. And these regulate their functions, they regulate their transport, they regulate their interaction with other organelles. And as mentioned, these serve key functions as dynamic hubs of lipid metabolism. Now, this is the story where the story, I would say, ends for most cell types in our bodies and tissues. But we have additional pathways in enterocytes and hepatocytes, in that they have a decision to make with their neutrolipid. They can also target this neutrolipid to lipoproteins for secretion throughout the body, where it circulates as VLDL or chylomicrons. And the loss or regulation of genes involved in lipid droplet biology results in many different diseases, particularly diseases associated with metabolic diseases like fatty liver disease, atherosclerosis, metabolic syndrome, where you accumulate lipid droplets. And so this is certainly the case in fatty liver, where you accumulate lipid droplets, and this can lead to eventual fibrosis and is a risk factor also for hepatocellular carcinoma. So we are very interested in addressing the mechanisms that govern hepatocellular lipid flux and storage in droplets. This was work from a previous graduate student, Alyssa Mathewitz, now a scientist at Panoplia Labs, and a current graduate student technician, Emily Maymond and Emily Torres, so Team Emily in the lab here. So we again turned to genetics to address this. In this case, we were performing a death-based screen. We performed a facts-based screen. We used Bodipi 493503 to mark lipid droplets. You can see here cells with basal lipid droplets, or which we've induced lipolysis or loaded with fatty acids. We can then sort these by sorting on a fluorescence activated cell sorter for low and high, and then do deep sequencing. Here we have a much more complex volcano plot indicating the genes that are regulating lipid droplet formation. On the right being genes that when knocked out accumulate lipid droplets, on the left being genes that when knocked out decrease lipid droplets. There are many genes that we expected, which gave us a lot of confidence in this screen. For example, DGAT2, the rate-limiting enzyme in the conversion of diacylglycerol to triacylglycerol. In addition, we identified the rate-limiting enzyme in lipolysis and its regulators. This enzyme breaks down lipid droplets, and as expected, when knocked out, accumulated droplets. Alyssa went on to do several screens. She wanted to understand how lipid droplets were regulated under different conditions, so we synthesized a new library based on her hits and curated lipid metabolic genes, and we did duplicate screens under 10 or 11 different conditions. And the motivation here was twofold. One, we wanted to understand the genetic modifiers under these different conditions. For example, fatty acid loading, oleate, palmitate, arachidonate, or different types of starvations or stress conditions. But we also, by performing these parallel screens, this gives us some amount of chemical genetic epistasis, where we can start to cluster these genes based on their functional profiles, and genes with similar functional profiles with related functions will cluster together. And this can give us some initial leads or clues about novel genes and what they might be doing. So I won't go through all of this complicated heat map here resulting from these screens, but I'll point out that at the very top of this heat map, shown in red, are genes that we categorize as core negative regulators of lipid droplets. So for example, if you knock out PMPLA2, which encodes ATGL or PMPLA2, the rate-limiting enzyme in lipolysis, as you would expect, no matter what condition, you accumulate more lipid droplets. We see very nicely that this clusters with ABHD5, its co-activator. For the remainder of the time, I'm going to focus on this gene, which clustered together with PMPLA2, CLCC1. At the time, we didn't know much about CLCC1. It's essential in mice and zebrafish. It's a polytopic membrane protein. It's annotated as a chloride channel. We don't think this is a chloride channel. I'll tell you what we think it does. It localizes to the early secretory pathway, as well as to the nuclear envelope. And there are mutations associated with some diseases. And we're interested in, and one of the reasons we prioritize this is that there are some rare variants associated with altered serum lipids and lipoproteins. This is one reason we prioritize this protein, in addition to the strong effects in its clustering with PMPLA2. So Alyssa went on to knock out CLCC1. These are in H7 cells. These are cultured hepatoma lines. You can see that in our control cell lines, we have some basal amount of lipid droplets. And in both of our knockouts, we get the accumulation of these larger lipid droplets. There are less of them, but they're much bigger. If we stimulate lipolysis by treating with an acyl-CoA synthetase inhibitor called trioxin C, in our control lines, we consume all the droplets, but these droplets are very stable in our two knockouts. At the time, we thought maybe that CLCC1 was a regulator of ATGL, a regulator of lipolysis. But we see that it's now that it's operating through a very different mechanism, which I'll tell you about in the next few slides. So these were in cancer cell lines. We also wanted to understand whether this was relevant to in the liver, in actual hepatocytes. So we've had a really terrific collaboration with Ana Paula Arruda here at Berkeley, as well as with Greg Koo at UCSF. We generated a floxed CLCC1 mouse, and then we injected AAV-Cre by tail vein to knock out CLCC1 in the liver. And this was a really striking result. My graduate student, I think, texted me this at like 11 at night. She said, "Oh my gosh, look at this." She had opened up these mice, and these are incredibly steatotic. So you can see this huge accumulation, this whitening of the liver. These are about 2 times the size. And this is just 4 weeks post-AAV, so there's no high-fat diet, feeding, or anything under these conditions, just CLCC1 knockout. I'll tell you more about this, but I wanted to mention that this was also observed and co-discovered by Xiaowei Chen's group at Peking University in a corresponding bioarchive, and also will be co-published together with Xiaowei's results. can confirm this in many ways using biochemistry, and I'll just show you some histology here. We see the accumulation of oil-red O stained lipid droplets, and I think what's really striking here are these EM images, electron microscopy, which we see all these lipid droplets accumulating in these hepatocytes in the CLCC1 knockout liver around this nucleus. So huge lipid droplets that are accumulating. So we wanted to get some clues into what CLCC1 was doing, so we fractionated cells, we floated them and did proteomics, and we were really shocked to find that these floated lipid droplets had low PLIN2. This is a canonical lipid droplet coprotein, but all these proteins that are known to be on lipid droplets were low. We could see this also very strikingly by immunocytochemistry, that PLIN2 wasn't just low on the lipid droplets. Usually it's stained here in green, it's on all lipid droplets. It seemed to be completely devoid from these lipid droplets. And when it can't localize to lipid droplets, it's usually degraded, and we see this loss of PLIN2. So we were really perplexed at this time. How do we have these huge lipid droplets, but they lack these canonical droplet proteins? And the answer came when we did additional EM. Here we're showing the ER. We have these nice contacts. The ER is surrounded by magenta with lipid droplets. But we see something very different in these CLCC1 knockouts. And this was a really nice EM micrograph. We have an ER sheet here, one of the membranes going all the way around these lipid droplets and coming back to that sheet. And what this is telling us is those lipid droplets are in the ER lumen. They've gone the wrong direction. This really explains why they're so resistant to lipolysis, because the lipases can't access the droplet because they're all in the ER lumen. And this also explains why we didn't see any canonical cytoplasmic droplet proteins, because these are in the wrong place. So we started to imagine how this might happen. The lipid droplet could be budding off the inner leaflet. We can also imagine that these are hepatocytes. They form lipoproteins, which look similar in structure to lipid droplet. They're just much smaller. This model gives us some predictions that they would be ApoB positive, and they'd require this enzyme, microsomal triglyceride transfer protein, MTP. So that's a testable model. So Alyssa went back to her cells. As you can see here, our knockouts are devoid of PLIN2 because they're in the lumen. If she treats with an MTP inhibitor and blocks the formation of lipoproteins, it reverses the flux of neutral lipids, and we get PLIN2 lipid droplets, which appear to be cytoplasmic and normal. Those lipid droplets that are accumulating in the knockout are ApoB positive, and we also have altered secretion of lipoproteins, both in our cellular model, looking at ApoB secretion, and in our in vivo model. This is just showing plasma transylglycerol, but we've also quantified ApoB and VLDL, and these are all massively reduced in the mouse model. So we have defects in the lipoprotein pathway and a really huge, profound shift in flux of these neutrolipids away from cytoplasmic droplets into stored neutrolipids. So what could CLCC1 be doing? We started to dig into the literature, and the only model we could see where some people have observed this type of phenotype was where they had knocked out TMEM41B or VMP1, two ER scramblases. They start to accumulate these large luminal droplets within hepatocytes. And so I emailed Tim Levine, a colleague, and I said, hey, does CLCC1 look anything like a scramblase? He's done a lot of work on these. He said, no, it doesn't really look like a scramblase, but when I do protein structure homology searches, it looks very similar to this domain, at least in this portion, to yeast BRL1 and BRL6. And we can see this here in this alpha-fold predicted structure of CLCC1, that it has this long helix. That's what we're calling a knuckle that's stabilized by predicted disulfide, this amphipathic helix juxtamembrane, and then this other helix, kind of at this very tilted angle. And this looks very similar to yeast BRL1. This is, again, an alpha-fold predicted structure, but it has these disulfide stabilized knuckle, this juxtamembrane amphipathic helix, and this other transmembrane domain within this domain. So we became very interested in what BR1 and BR6 do. And these have been studied by Karsten Weiss's group at ETH Zurich and others. And what they had found is that BR1 and BR6 cooperate to mediate membrane fusion, at least this is the model, for insertion of nuclear pore complexes. So here we're showing the model of what BR1 and BR6 are thought to do. They're thought to accumulate here at these sites where you have the nuclear envelope, the inner and outer membranes, and eventually in order to get a nuclear pore, you need to fuse those membranes to get a hole in the membrane. And these localized to these sites and are thought to mediate the fusion for insertion of these nuclear pores. And so in the absence of BR1, these are essential in yeast, but you can deplete them acutely. You no longer fuse the membranes and you get these large nuclear blebs off the nucleus. And this is what this looks like. This is from Karsten Weiss's group. You get these large nuclear blebs off the membrane because you've no longer inserted nuclear pore complexes. So we went back to our EM micrographs. I feel bad that we missed these. Initially, we were so focused on the lipid droplets. But when we zoomed in here on the nuclear envelopes, we saw these remarkable nuclear blebs accumulating on the nuclear membrane. We can also see these blebs or herniations within our knockout mouse liver, as well as with fluorescent markers of nuclear herniations or blebs. This is a marker from our Christian Schleicher at Yale. We see this very nicely on primary hepatocytes from these animals. I'd like to point out that since we posted this bioarchive, several groups have posted preprints in the last year confirming these findings where they knock out CLCC1 and accumulate these nuclear herniations. And so we wanted to study this in more detail, and together with Ana Paula Arruda, we looked more closely at the membrane and organelle architecture changes in these cells. These are in the mouse liver, so on the left, the wild-type liver, on the right, the knockout liver. Of course, you can see the striking accumulation of these enlarged luminal lipid droplets, these altered lipoproteins shown in yellow. So these cells are just packed full of these. But as we zoom in, you can see that there are other membrane architecture alterations. The ER in cyan appears more vesiculated or fenestrated. But what's really striking is the nucleus. So you see all these nuclear blebs or herniations around the nucleus. And this kind of dent into the nucleus is a really large lipid droplet just pressing in and deforming the nucleus. Since these are serial sections, we can walk through and we see these structures. So we can also show that this has the corresponding reduction in nuclear pores as expected, consistent with our model. And so I'll just end here by concluding what I've told you about today in two parts. In the first portion, I told you about our discovery of FSP1 and its role in protecting membranes, both the plasma membrane and ferroptosis, as well as neutrolipids within lipid droplets, and our identification of FSN1 as an inhibitor. And the second, I told you about CLCC1 and its role in neutral-lipid flux, and what I think it's probably its ancient role in mediating membrane fusion for the insertion of nuclear pores. When you lose CLCC1, you accumulate in large luminal lipid droplets. You no longer insert nuclear pores through this pathway and accumulate these herniations. I'll end just by thanking all of the members of my group. I tried to point everybody out along the way that did the work, as well as our wonderful collaborators. Also, a big thank you to the funding agencies that helped make this work possible. And I'm happy to take any questions. Thank you.
Thank you very much, James. Terrific presentations. Okay, please go ahead.
James, I just had the question on FSP1. It's actually Patricia. I wrote it incorrectly. I don't know if this is possible. Have you tried overexpressing FSP1 and what happens in those cells? I don't know if this is possible.
Yeah, we have overexpressed FSP1, and these cells become very resistant to ferroptosis. We can also overexpress FSP1 targeted to different membranes to kind of uncouple its role, for example, at the plasma membrane or at lipid droplets. And targeting it to the plasma membrane is sufficient to block ferroptosis. In the knockout cells, we can also target it to the droplets. This is sufficient to protect the stored neutrolipids. So we think that it's playing roles at different membranes within the cell.
Okay, interesting.
Thank you. Yeah, go ahead, please.
Hey, Jim. Padhu here from Indiana University. Beautiful talk. I have a question regarding if the if whatever you see in hepatocytes, is it across tissues or you're seeing it only in hepatocytes, like the accumulation of lipids in your CLCC1 knockouts?
Yeah, thank you for that question. We think the lipid droplet phenotype seems to be hepatocyte specific. So if we knock this out if we knock it out in HepG2, for example, we see the same thing, but knock out in U2S or other cell lines, we don't see this. We do always see the nuclear pore phenotype in these nuclear blebs, and we think this is probably why CLCC1 knockout is embryonic lethal. And we think that this is its conserved and ancient role. We imagine that maybe we'll also see something in enterocytes, which form chylomicrons, but probably it's picked up this moonlighting role in regulating neutral lipid secretion in hepatocytes and maybe in enterocytes, if I speculate.
And if I can ask another question, what happens to other perilipins?
So in these cells, in hepatocytes, we no longer get them on the floated droplets. Okay. I'm imagining that they're degraded. So not all of the perilipins will be degraded, but perilipin 1 and perilipin 2, if they don't insert into droplets, are often unstable and degraded by the proteasome. Some of the other perilipins can exist in cytosolic states, like perilipin 3, maybe.
I'm thinking about the oxidation, beta oxidation of these lipids when they get accumulated. What is the response? Like, how is it with the contact site with the ER or mitochondria?
Yeah, that's a very interesting question. At least these luminal droplets seem to be completely spatially sequestered. So we can, for example, overexpress ATGL in these lines, which usually is really powerful inducer of lipid droplet lipolysis, and these droplets are protected. So I think because they're in the lumen, in this early kind of lipoprotein biogenesis state, they're protected from lipolysis. And whether there's any flux between the cytoplasmic and luminal spaces, this I think is really an interesting question that we're interested to answer.
Okay, beautiful talk. Thank you.
Yeah, thank you.
I have two questions from Daniel Haas.
Yeah, they're pretty brief. The first is, does FSP1 have a preference between NADH and NADPH? I'm noticing you kind of used NADH in some of the assays, but here you have maybe either or.
Yeah, in vitro it can use either. So I think in vitro we tend to just use NADH, but it'll use either. I think we've seen some slight differences, but it usually can certainly use either. I think in cells, I think this is an open question as to which of these pools it employs. There is a recent paper suggesting that it employs membrane-associated pools of NADH, which I think is very interesting, but how these NADPH and NADH pathways are kind of regulated and feed into the FSP1 pathway within cells, I think is an interesting and still important question.
All right, cool. Second question, very quick. I think I saw earlier in one of the slides that there's a retinitis pigmentosa phenotype in the CLCC1 when you have a mutation in this. Is that right?
That's correct, yes.
Is there any visual phenotype in your mice?
That we haven't looked at. So in our mice, we've only knocked it out in the liver since it's embryonic lethal with the full knockout. So we've started to cross this mouse with some other drivers to look at its role in other tissues. There has been work from others with mutations. For example, there's a spontaneous mutation that arises in mice that causes an ataxia phenotype and neurodegeneration. There's another group who has looked at this in motor neurons using a direct knockout, and they get some interesting phenotypes there. But the mutation in the retinitis pigmentosa is in this near the end terminus of the sequence, and I think I'm trying to remember now, but I feel like it was a surprising mutation, like a glutamate to aspartate or vice versa, that has been implicated in this disease. And how that's related isn't clear. Yeah, but we haven't directly looked at this.
All right, thank you.
Thank you very much. Maybe I have my chance to ask a question at the last minute. concerning, let's say, like the role of lipid droplets in ferroptosis, yeah, there is a lot of research in this way, right? And what is your take in terms of which DGAT isoform case plays a key role? Is
Yeah,
it
yeah,
due to DGAT 1 or 2? Or?
We haven't really, I would say, like, we don't have a definitive answer on our end. We've tended to, in our studies, at least in the cell type, under our conditions, when we inhibit with one or the other, it's not a full loss of droplets. So we just inhibited with both to get a clean answer. But I think that's a very interesting question because they're very different enzymes, DGAT1 and 2. They're kind of converge on their activity, but they're very different in that DGAT2 is a hairpin protein that potentially can traffic from the ER to the droplet, whereas DGAT1 is this polytopic ER membrane protein with a wider substrate clientele. And they've been implicated in different activities, like DGAT2 with maybe more with growth. We've seen DGAT1 maybe more playing a role in mitigating lipotoxicity. But in this case, I don't have a good answer for whether one or the other will come out as more important.
Yep, that's the interesting question, which also worries us a bit. Yeah. Okay, the very last question.
Yes. Hey, James, a quick question. My question is, where does the phospholipase go when you inhibit the lipid droplets formation?
Yes, I feel like we've done this. That's a good question. I'd have to double check to look at the lipidomics. The thought is that that's going to channel into phospholipids, but I'd have to take a look to confirm that. But that was our thought, was that the blocking of the neutral lipid synthesis, we'd see flux there into the phospholipids, but I'm not sure if we saw that. But the idea there is if it is channeling into phospholipids, that we have two parallel pathways to protect membranes from damage. So loss of FSP1 under those conditions, even though we would have more PUFA in the membrane, they'd be protected by GPX4, whereas the neutral lipid in the droplet seems to be kind of heavy reliance on FSP1, if that makes sense.
Thank you.
And Jason Miller?
Thanks. Thanks again for the amazing talk. The history of trying to supplement CoQ for mitochondrial disorders, et cetera, has of course been quite poor. And everyone sort of thinks it doesn't necessarily get to the targets appropriately. But I'm curious if there's a stoichiometry issue where FSP is limiting, and whether or not you have any data about, that FSP enhancement is the key missing factor, not the actual sort of bioavailability of CoQ.
Yeah, I can imagine that we'd probably run into both of those issues. Certainly the delivery can be challenging for CoQ, and there have been some interesting papers on delivering CoQ precursors, at least for some genetic diseases associated with that pathway. FSP1 in certain cells certainly can be limiting. So cancer cells kind of have differential expression, and this is one of the first findings that kind of hinted towards an effect in ferroptosis, was a strong correlation in expression. So there's certainly cancer cells with lower FSP1, and if we add in more FSP1, it can drive ferroptosis resistance, indicating that it's limiting, and at least under those conditions, its substrate is not the limiting factor. But those are in cultures, so whether in vivo, we run into similar situations, I think that's an open question still.
Thanks.
Thank you, James, very much. That was really great talk and very inspirational, like always. And thank you all for joining today, and bye-bye.
Thank you all. Happy holidays.